Good evening, everyone. My name is Takiwa T. Smith. I'm founder and executive director of Science, Engineering, Mathematics Link Incorporated, and I want to welcome you to our first Team Science Cafe of the 2021-22 program year. I am so excited because this program year is our 17th fiscal year. So this is our 17th year of exposing you to STEM and STEM careers. And so the Teen Science Cafe is a program of our Math and Science Career Academy, which focuses on um, exposing kids to STEM careers, hands-on STEM activities, and interactive, let me slow down, demonstrations. And we focus on the Teen Science Cafe in teens because this is the time in your educational journey where if you're in middle school, you're making decisions about high school, and in high school, you're making decisions about college. And we want you to make informed decisions by meeting people that do things that you are interested in. And so that is why we host the Teen Science Cafe. And we are still hosting it virtually because I know things are quote unquote getting back to normal, but they're not. We're still in the midst of a global pandemic, and we want to keep our staff safe, our volunteers safe, and you safe. So, without further ado, I'm going to bring our program coordinator, Carlin Pounders, on so she can introduce tonight's speaker and our topic for this evening.
All right. So, hi everyone. As Ms. Tequila Smith just said, my name is Carlin Pounders. For those of you who are tuning in for the first time with us, so happy for you all to be here. Very excited to share that we had a number of you sign up. We've had this is the second highest attendance that we've had for our virtual events. So I hope that all 65 of you are here with us tonight and ready to hear from our speaker. So tonight's speaker is Nella Delva. She is a fourth-year PhD candidate in Biomedical Sciences at Florida State University. She is a neuroscientist in training studying mood regulation, particularly major depressive disorder. Delva has worked at H. Lee Moffitt Cancer Center in Tampa, Florida. She was also an adjunct professor at Hillsborough Community College, also in Tampa. Now, via STEM Amazing Advancement, she continues to mentor, coach, and teach minority students in order to encourage and expose them to STEM fields as a potential career path. So there's some other exciting things that about Nella that she is going to share with you. So without further ado, I will hand it over to her.
Hello, everyone. So thank you for your attendance. Let me get my cursor right. So thank you for the nice introduction. I appreciate it. So I'm hoping that everyone can hear me correctly. So today's discussion will be about mood, specifically about neuroscience as a neuroscience perspective of moods. Some of the learning objectives today, I will be describing my academic journey and goals. I'll also be discussing some hands-on research experience that I've had along the way, while stating some of the importance of biomedical research for clinical development and advancement. And together we'll take a look at different neurotransmitters and how they are affected when we're happy and when we're sad. In addition, I will discuss what happened when the neurotransmitters are not doing so well, and that's what happened in depression. Also, I think it's important to discuss real-life example of biomedical research, which I will discuss my research, the research that I'm doing today, And I'm saving the best for last. So I will share with you a real sheep brain, virtually, of course, and we'll dissect it together. So a little bit about myself. I was born and raised in Haiti. I permanently moved to the wonderful land when I was in 11th grade. Therefore, my academic and research journey was all completed in the state. Secondary, I love science. This is all me here. Particularly, I love medical research. I'm specializing in neuroscience, and I will be discussing that a little bit further. Thirdly, I have a passion for STEM advocacy, especially for minorities. There's not enough minority in STEM. We all know it. So my overall goal is to be a part of inspiring students to consider a STEM career. Consequently, I'd love to be part of helping raise the current statistics. So my academic journey is more like this meme right here. That's the best way I can describe it. The top panel, this is me after high school. I thought life would be a straight line. I wanted to go to college, then grad school, and eventually have a dream job and happily ever after. But things turned out more like this bottom panel right here. There was a lot of challenges. So there was some rocky path, and I had to take a bridge somewhere. I was stranded in some type of island. I don't even know how to swim. I had to go up a mountain. I got wet somewhere. So overall, it's all figuratively, of course, but overall, my point is the path was not easy. So let's talk about more in detail in a chronological order about my academic journey. So I graduated in high school in '07 with honors. I was part of a biomedical environmental advancement magnet program. I just had at this point a simple and naive interest in science. That was all. Then I wanted to have some time to explore. So I was advised that community college was the cheapest place to do it. So I went to community college. My interests there changed a lot. I changed to different career paths. I knew I didn't want to go to medical school, but also I knew I loved science. I didn't really know what I wanted to do. So I went on and transferred to a four-year institution, majoring in biology. Barry University really was where I narrowed my interest a little bit more. I was accepted to this minority biomedical research support program, which drastically changed the course of my academic career. After graduation, I decided to continue doing biomedical research, doing a postbac program at University of South Carolina in Columbia, which consequently resulted in me working on a master's thesis. I acquired a master's degree in biological sciences in the same lab. Then after my master's degree, I thought, it was time to get a job doing what I loved. So I worked at H. Lee Moffitt Cancer Center doing research, preclinical research, while I was teaching at a community college there. My story still is still being written because I'm now a PhD biomedical science program at FSU, training in neuroscience, specializing in neuroscience. So one of the most asked questions I get is whether biomedical science is like medical school. So the answer is no. By definition, biomedical science is the application of principle of natural sciences, especially biology, to clinical medicine. So in other words, medical school deals with patients, their symptoms, and provide them with a plan for disease prevention and also appropriate treatments. Excuse me. While biomedical sciences deals with the unknown of clinical medicine. For example, we deal with the unknown about cancer. We try to answer important questions that can help us find the cure for cancer. We also deal with the unknown about neuropsychiatric disorders such as depression. We question why are some people more likely to be depressed than others, and how can we find better therapeutic treatments for these diseases. Now in COVID era, biomedical scientists are really essential to find out things about the virus because the virus was pretty much unknown. And there were also very essential to trying to find ways to defeat it, which led to effective vaccine that we have today. So I really like this meme because it shows an accurate depiction of how we're seen as scientists in movies and in the media in general, even by our families and friends. In the media, a scientist is depicted as a white, nerdy male, and the movies were depicted as a sci-fi experiment in the basement, or even in the basement doing some illegal drug related. Some people think that we're growing ears behind mouse backs, or even coming up with like explosive chemicals. But a lot of the things that we do as scientists is these two. We do a lot of analysis. we we do a lot of wet lab work, of course, a lot of brainstorming, a lot of collaborating. So that's really what we do. Um, so this is me in the lab. I do, as I mentioned, wet lab work. I work with solutions here and making solutions. I also work with mice, testing for different therapeutics. These are the different cages of my mice. I check on them every day because I breed mice and they do have pups. You know, I have to count the pups and make sure that I wean the pups. And also, I do a lot of analysis in the microscope, looking at gene expression profiles. So I'll talk about that in the next few slides. So once I complete my degree, I have several career options. I can work in improving tools and techniques for preclinical research. I can study healthy biological processes, or I can even continue doing what I do now, studying causes and progress of diseases to find possible treatments for them. So you may wonder, how did I even get started doing research? A lot of people ask me that. Well, I really didn't know about research when I was in high school. Um, but being accepted to the biomedical and environmental magnet program was really key. I did study a lot. This program exposed me to advanced biological courses, which gave me a strong foundation for science, really. It wasn't until really my sophomore year in college that that strong foundation in science that I had was converted to doing wet lab research with my genetics professor. So my genetic professor saw potential in me and she invited me to do research with her. So we did research using zebrafish. So we were like exposing the zebrafish in ethanol, looking at their brain and how their neural circuits was changing in different ethanol concentration. And then I went on and did a summer research internship with that same genetics professor that opened up the world of genetics to me. So I started doing research in genetics when I was at University of Missouri during that summer program, and then I continued on in my undergraduate doing genetics work with my professor. I learned a lot during this experience. I learned how to manipulate genes, how to shut down genes, how to overexpress genes, and how to do it, where to do it, and the different equipments that they have that can effectively get it done, not only in zebrafish, but also in human cells and mice and so on. So I moved on doing more research in genetics because I really liked it. I did genetics and proteins and looking at modification and human cells this time. And then I went on to studying human cell even more, but in my master's thesis, I went and studied virus. So I do have a background in viruses. I study how virus is affecting human cell, especially HPV virus, because it pushes human cells to become cancerous over time. So I was studying how does it do it and how can we stop the virus from doing it. Then my genetic journey opened up many doors. I was able to work at the Cancer Research Institute that I mentioned previously, working with animals, actually with mice. And what we would do is use human cancer cells, inject them into mice, so the cancer cell will grow in mice. And then we will test, this would be a different therapeutic agent. So we will test different therapeutic agent. I'm hoping that this therapy would help decrease the tumor size. And we were successful. This project was later approved for human clinical trials, and then it succeeded to be an effective treatment for human uveal melanoma. During my research, I wanted to shift and use my genetic background to study mood regulation. Specifically depression, because it's such a hot topic nowadays. But we will go over what I do a little bit further. So I hope that the past research experience that I just went over gave you a small hint of what biomedical research is really about. Overall, biomedical research is subdivided into many different groups. In my graduate program, we have molecular, cellular, developmental biology, which studies the heart, and different spinal injuries, viral infections, behavioral and system biology, which is basically neuroscience, because I study the human behavior, psychiatric disorders, diseases, and genomics, and so on. In addition, we have a biochemistry and biophysics subdivision, where you can use biochemistry to come up with targets for diabetes and tissue damage and repair, and so on. So, of course, I decided to go with the specialization of neuroscience because I knew I could use my background in genetics to study human behavior. And I also wanted to be a part to find better treatments for neuropsychiatric disorders. For those who don't know, let's dive into what neuroscience is. So neuroscience is a study of and function of human brain, really, as well as the nervous system. So neuroscientists I use cellular and molecular technique to map the brain at a mechanistic level. So that's what neuroscience is. Since we can't open a human brain to study it, because that would be abuse and violation of many moral code of ethics, obviously, we use rodent models, specifically mice in my lab, but you can use voles, you can use rats, and other type of rodents. There's many different labs that use different types of rodents in my institution. So what a lot of other people don't realize is that human and mice genomes are actually 85% identical. That's what makes mice so attractive to scientists, because if you're studying the gene and then a mice has a gene that's 85% identical to humans, you want to study the gene in mice. And that's only an average. Some genes are 99% identical to humans. Some genes are 60% identical to humans. The gene that I'm studying is about 90% identical to humans. Makes it even more easy. So also, a lot of the diseases that we study in humans, we can study them in mice. They are similar to genetic disorders in mice as well. So in order to study their development to come up with therapies, the mice helps with that. And overall, the rodent model helps us just coming up with things faster, because when you study genetics, you also have to study different generations, and the gestation for a pup and a mice is only 20 days. So it makes it much faster instead of 9 months in humans. So what do we do with the mice? Well, this is a cartoon representation of what we do with them. We have the mice here. We obviously want the brain. This is a cartoon representation of the brain. This is the front of the brain. This is the back of the brain. We slice them so we can use the brain slices to look at neurons. So how do we exactly do that? You may ask. Well, unfortunately, the mice have to get killed. And the heads, it's a little drastic, but we do chop off their heads to collect their brain. There's no other way to do it. We say a little prayer. I say a little prayer to help them through it. And then we use a brain slicing machine to slice the brain. And then because we're looking at neurons, we're looking at the cells, the brain cells, the different genes are expressed in different brain cells. We can't do that looking at the slide at the naked eye. We have to use a microscope to do that. And that's what's shown here. So what else can we look at under the microscope? Well, we can also look at different molecules. So one of the molecules that we look at with a specialized microscope is neurotransmitters or molecules. They're very, very, very small. So this is more of an in-depth of where those molecules are. This is a brain slice. Here, there's one brain in purple. I'm sorry, one brain cell in purple and one brain cell. Is this blue or purple? Okay, blue and purple. And then this little square is showing you what is shown here. So those circles are neurotransmitters. Those circles shown right here. They're neurotransmitters. And what are neurotransmitters? Well, just think about neurotransmitters as a signal between one brain cell to the other. So those signals are basically shown here in those little dots. They're not actually dots, but they're shown here in those dots. And the way that the neuron communicate is basically through this. The more they release those little molecules, the more that they translate a message. A message is translated basically. So those little molecules of neurotransmitters, there's many different shapes. They come in many different shapes and sizes, not just one. You probably have heard of some of them. They are called dopamine, serotonin, adrenaline, glutamate, endorphins, etc. So this is basically the chemical structures of those neurotransmitters shown here as a representative of dots. So dopamine is also referred as a pleasure neurotransmitter. So it gives us the feeling of pleasure and motivation. And also it's very important in addiction because you're only addicted to things that you find pleasure and motivation to get, right? The other one that's really talked about is serotonin. So it's a mood regulating neurotransmitter. It makes us feel at ease and at peace and happy. So also a lot of people don't know is that this same neurotransmitter regulates sleep and then the digestive system. So when you're not happy, you don't really sleep. When you're not happy or you're not feel at ease, some people cannot even eat when they're not happy. The reason being is that if the neurotransmitter is not there, they can't digest and therefore they can't eat. And some people overeat because if the neurotransmitter is dysregulated. Adrenaline is one that's being talked about a lot and is responsible for the fight or flight response. Really, this neurotransmitter is produced in stressful situation, also an exciting situation because you do freeze up when you're excited. So just imagine it this way: if you're on a hike and all of a sudden you see a bear right in front of you, this neurotransmitter prepares your body for escape. So that's why you freeze up because it wants to increase your heart rate, increase the blood flow, and gives you a boost of energy so you can escape. So that's the main function of this neurotransmitter. Also, another one that I hear a lot about is glutamate. But glutamate is responsible for memory. So whenever you're learning a new task, you get a lot of boost of glutamate in your brain because it helps you make connections and stuff like that so you can remember the task that you're learning. So I wanted to take some time to address these three neurotransmitters that's being talked about a lot in the media. And there's also a lot of misinformation out there. So I wanted to clarify them a little bit. Dopamine is always referred as the feeling of being in love, the love neurotransmitter. However, it's more accurately referred as a reward molecule in the scientific community, at least. So some of and anything as I don't food, um, drugs, and even sex can be seen as a reward. So if I can give an example, I love chocolate, but that's because it's associated with a reward. Just think about it when you're a child, whenever you're able to eat sweets is when, you go to a party or you're at the pediatrician office or you were good that day and they give you a cookie. So that's an association of a reward. So that's why I love chocolate because I associate it with something that I did good. I'm, rewarding myself. So if I was living in the world where there's plenty of chocolate and I could get no cavities and I could just eat as much chocolate that I can, possibly I would no longer associate chocolate with a reward and therefore I would no longer love it. So that's the idea of dopamine. Serotonin is often talked about with regulated mood and being a feeling of happiness. That's accurate. You do have a feeling of happiness when you have a boost of serotonin. Oxytocin, however, is another molecule that's being talked about a lot because it's associated with the love chemical that's being seen. However, it's really associated with pair bonding. And that's important because whenever we're having a romantic relationship with somebody, we want to be attached to them. We don't want to be associated with them. We want to bond with them. So that's a molecule that is very strong in helping you bond. But it is not only and partner relationships. Early attachment between mothers and infants is increased as well, and it's also increased in friendships. So if you have a best friend and you feel very connected to them, and during developing this bond with this friend, you have a boost of oxytocin to help maintain that. Um, so Glad I clarified that. So what happens when you're happy? Well, when you're happy, you have a boost increase of serotonin. So this is basically a cartoon representation of the quantification of happiness and anxiety in terms of like the different neurotransmitters in different colors. So when you are happy, you have a boost of serotonin levels. That's why you're happy. What happens when you're in love? So when you're in love, you get a lot of different chemicals. And that's important too because we have a boost of dopamine because dopamine, as I mentioned before, makes us feel rewarded. It's a reward molecule. We'll feel like we are being rewarded to find this partner, to have this good friend, the infant. So it's like a reward molecule. Serotonin, again, is about happiness. Oxytocin level increases as well because that's the pair bonding molecule that helps us being bonded with one another and being attached strongly to one another as well. On the other hand, low levels of dopamine and serotonin is really not good. It makes us anxious and it makes us depressed. Also, I want to bring up your attention to schizophrenia because you would think the more chemicals you would get is the best, but apparently in schizophrenia you get too much of dopamine. So if you get too much of something, it's not so good for you. It gives you a disorder. So just wanted to make sure that I mentioned that. And there's this other fight or flight response. You get a boost of adrenaline and noradrenaline for these two. So I want to mention depression because, it's something that is being talked about a lot. So the major depressive disorders is really a very formal name for depression. And really it's cause of a chemical imbalance in the brain. So in a normal person, and these dots represent the as I mentioned before, these are cell 1 transmitting a message or a signal to cell 2 right here. But these dots represent serotonin, just to be specific. So in a normal person, you have a boost, quite a bit of serotonin here. However, when you're depressed, there's not much serotonin. So a lot of the treatments out there that are available is responsible to making sure that the cell continues to provide serotonin to give the next signal to the next cell. Remember, like, think about it as a phone signal. If I'm trying to talk to you and then you can't hear me, or you hear some stuff that I say, but you don't hear everything, it's a problem. So that's where the depression comes in. But if you put an amplifier for the signal, for example, which is the yellow dot seen here, then it's better. Then you're happy again, and you're not depressed. So that's what the SSRI treatments do. They're amplifying the cell to make more of serotonin. So I want to share with you some of the work that I'm doing in my lab. Of course, I focus on depression. So I'm sure you all remember that, the peak of COVID during the lockdown, many of us felt like we were depressed. I felt like I was depressed. So just to say that depression is something that continues to affect the community. And no one is really exempt from it. It affects all age group. 1 in 6 people in average is affected every year. And just I wanted to also clarify, when we're talking about depression, we're talking about symptoms that are identified for a person with experiencing depressed mood, and loss of interest and pleasure for an extended amount of time, not just a couple months, but we're talking about 6 to 8 months. And then it seems like the person is not doing better. So yes, depression doesn't mean you feel bad today or this month or next month. It means that you feel bad for more than 6 months. So most of you don't know this, but in addition to environmental factors, depression has genetic contribution as well. Yes, you have genetic contribution to depression. And then one of the important things that's causing depression is stress, chronic stress. So constantly being exposed to stress day after day after day after day. So the neurotransmitter dopamine is a main neurotransmitter that I'm focusing on right now. And the reason being is that it plays an important role in modifying the stress level that we internalize. So scientists such as myself are like, wait a minute, maybe we can look at dopamine as a form of prevention. So can we use it as a therapeutic target? Can we take it out? So I looked more into it. Thankfully, we found some old data mapping both human and rodent models, so models of mice, pinpointing different cells in different brain areas, as you see here, like different areas right here where dopamine receptors are expressed during development. That's important because you don't want to start by looking at dopamine in the whole brain, because that would be an issue when you are looking into making a target for diseases. You want to pinpoint where exactly you want the dopamine, take out the dopamine, if the dopamine is an issue. So this is a picture of the actual cells that is an issue. So those of cells are specific because possibly taking out dopamine in those cells can be a therapeutic target. People will be less likely to be depressed, and be more resistant to stress at the same time. So the good news is we did it in mice. We deleted dopamine and those group of neurons located in that specific area I talked about, and the mice were more resistant to stress, and they were less likely to be depressed. So basically, where do I go from there? I continue doing my research, trying to identify other underlying physiological changes in mood disorders, specifically depression. What is those other things that I'm missing here? Because it does work in mice, but we're talking about human beings here, so I need to have all my bases covered. And the overall goal is really to develop a potential strategy so we can have a unique therapeutic to treat depression or even prevent depression if we can. And that's it. The last thing I wanted to do before we leave today to allow you in my world where we will all be dissecting your brain together. It is a sheep brain. The reason being is that it's bigger. The mice brain is too small, so the camera wouldn't catch it all. So before I do this, I think I'd like to take some question if there's any in the chat, of course.
Let's see. If anyone has any questions, please drop them in the chat and I will read them out. So this is a picture that you took.
No, I did not took this picture. No, I would like to say yes, but it was not me. But it does look like that. It's in a solution right now, so it doesn't look like that. Ooh.
It looks super high resolution. Okay, it looks like we don't have any questions right now.
Oh, I did notice a lot of comments in here. Are you able to see this?
Yes, someone just asked, what is your favorite thing about neuroscience?
That's a very good question. I think my favorite thing about neuroscience is when I see how much gene, when I, let's say, knock out a gene or take out a gene, and how much it affects the mice behavior, because you can tell it right away once you knock out that gene. It's just seeing like how its genetics translate to behavior. That's my favorite thing, really. Also, the hope of making a difference and finding this, new therapeutic treatments for diseases that have been an issue for a while. So that's my other favorite thing.
Yeah, but it's like life-changing for people, the type of work that you can do in neuroscience. Another question is, was neuroscience always your first choice? I found it really interesting that you talked about working around cancer research and how you went from doing the cancer research to doing research around depression.
Yeah, actually, that's another good question too, because it's a full circle for me. When I was working with the zebrafish with my genetics professor, we were actually doing biological neuroscience and the zebrafish. So we were looking at different brain circuitry that were affected with the gene that we were knocking down. So I just happened to stumble upon doing cancer research because I was working in genetics. Because genetics, you can really go any places with that. It's very, very flexible. So, yeah, I mean, I find it more fascinating now than I was than before. But yeah, I decided to just switch gear and see, you know, looking at behavior because I thought it was like an interesting aspects. But that happened when I was working in cancer as well, because as I was working in cancer and looking at different therapeutic treatments, I noticed how much chemo was affecting the patients, because they were more likely to be depressed after chemotherapy. And after certain therapies or radiotherapy, they had very, very severe depression symptoms. And that's when I started asking myself a question saying, wow, I wonder what is it about chemo or cancer treatments that makes, is it just the fact that they're going through a lot of stress with this disease, or is it any other genetic factors that is changed due to that? So that's kind of what geared me towards the world of neuroscience overall.
So you mentioned genetics, and that relates to another question that we have, which is how can depression be genetic?
Yeah, a lot of people don't know that. That's yeah. So it's because we have genes. All cells in our body have genes that regulates the expression of proteins and regulate the communication to other cells. So depression is genetics because if we have a neurotransmitter that is not being produced or is being produced too much, and the brain cells, um, we can be more likely to be depressed. So remember how I said that serotonin was more of a happy um, molecule. So let's serotonin is produced by gene. So that gene, if it's affected, if you inherited a gene that's dysfunctioning, you can't produce serotonin. So now you're more likely to be depressed because you don't have the happy molecule being produced in your brain cells. So that's in the sense that makes that's one of the reasons why some people are more likely to be depressed than others, because they may have a dysfunction. They're just not, lazy or anything like that. They may have a legitimate biological disorder that doesn't allow them to be as, bouncing back as much as you. They might have a dysfunction in their genetics where they don't even produce the happy molecule that I talked about.
Okay, so we're gonna ask, yeah, we're gonna I'm gonna share with you this last question so that then we can move on to the demonstration. So the question asks, are most of the medications prescribed to people serotonin analogs? And also, are you aware of any natural compounds or diet approaches that people have tried?
No, to the first question, trying to think about what was the first question is no. Um, some of the medication or dopamine antagonist or agonist, because sometimes um, some people are resistant to serotonin treatments, and that's one of the reasons why scientists now are gearing towards looking at dopamine, because all the current treatments that we have are serotonin targeted. And then a lot of the people who are taking those are like, I don't feel anything. I don't know. This is not doing it for me. But some people take it and they're like, oh, I'm fine. Um, so yeah, there's a lot of different targets out there for different neurotransmitters. That's now there's a lot of new therapies that target different things for the same disease. That's one of the reasons why they say if that doesn't work for you, tell your psychiatrist that they can put you in something else. And if the next thing doesn't work for you, tell them so they could put you in the next thing. So everybody's unique. With genes, there's not a way to calculate this is going to be the best for that person. We're all unique in our own way. We might be resistant to serotonin, dopamine might work better, and glutamate might work better. So it all depends. What was the other question? I think I missed
The
that.
other question was, are you aware of any natural compounds or diet approaches that people have tried?
I am not aware of any current literature of natural compounds. I do try natural. They're not like tested by scientists or anything, but one thing that I've tried that I feel like it worked for me was like ashwagandha. Ashwagandha. I can't even say it, but it does. I feel like, and that only saying this because I'm a neuroscientist in training, that it's a serotonin targeted agent because it makes me so at ease and calm and makes me want to go to sleep. So there's a lot of natural remedies that other people or nutritionist says is going to do this for you, do that for you, although they're not supported by scientists, or scientists may or may not have looked at them. I don't know any studies as of now. There might be out there, I just don't know of any right now. But there is, like I said, ashwagandha worked for me, and that could be just the neuroscientist in me, just knowing like the way that it worked for me. I'm like, oh, this is a glutamate, not a glutamate, but a serotonin targeted treatment, because I felt very at ease.
So we'll move on to the demonstration, and then we'll come back to our last question.
All right, so I'm going to move around here because my camera is not working as it should. So we'll go to my lab. Oops. I'm hoping that you guys can see. Oops. Well, you might not be able to see me, but I hope you can see the tray that I have here, right? It's perfect. All right, so we'll go over I think this is enough. This is our sheep brain. It doesn't quite look like the picture that I sent you. But I hope you guys can see it. It's pretty. This is the brain. I talk about a picture where you could see it like this. Let me turn it around. This is the top of the brain. This is the bottom of the brain. This is the cerebrum, and this is the cerebellum, and this is a brain stem. So this little part right here is responsible for a lot of involuntary movements. And then the big part, of course, is responsible to all the analysis, brainstorming, planning that we do. So I will show you guys where in my research that I focus in a minute. So once I dissect it, but to look at the bottom, like right here, this is the olfactory. This is for where all the information in your nose come to, is like this little area right here. The ocular, all the information that you see comes right here. There's a couple of veins right here that you can't really see, but it's right here. And then the hypothalamus, you can't really see it, is also responsible for involuntary movements that you can see in the bottom right here. So we'll dissect the brain, and I'll show you what area and where the cells come from that I focus on in my research. And take this. This is like a lab knife. That's not the knife that you cook with. So I'm going to cut it right here to show you a better view. I think you guys can see it. So I'm going to go this way. I'm cutting the brain. This is a left hemisphere, and this is a right hemisphere. Cutting it. So I'm going to take the left hemisphere right here. It's a lot of, okay. Everything is disposable, so I'll be throwing everything away once I'm done. So the area that I focus in my research is this one right here. So cells in this area during development, they kind of migrate, they move. And once they move, they go all around the cortex right here. So those cells are the one expressing dopamine, and then once they express dopamine, they're more likely people that have those expression of dopamine in those cells are more likely to be depressed. So what we do is once we delete dopamine in those cells, we scan those and we look at the expression in those mice right here. So again, those cells, remember this is an organ. Organ is have it's a trillion number of cells. Those my focus is from cells that are derived from this area, and then they migrate during development. So we do adult, they migrate during development and being in the cortex right here. Also, once we put them in the slide, we cut, as I mentioned, we have like a brain slicer. We cut, we're more interested in the middle, the front middle area section, which is where those cells are expressed, because they're expressed right here, because they migrate right here. They don't migrate all the way in the back, so we don't worry too much about the back section of the brain. But we do cut the brain right here in slices, like a pizza.
Um, Nella, I have a question that somebody else may think too. It
Okay.
all looks the same. So how do you know what part? Is it that because you're a to me it all looks the same. So how do you know what pieces of the brain that you're cutting?
Yeah, so it's something that I had to get used to. It looks right now because it's been in a solution that is a preservative, so it kind of like bleaches the brain. You can't really see the different areas very well because it's not a fresh brain. But when I do have my mice and I collect the brain, they're fresh brains. So I
Okay.
mean that, as soon as they die, I cut their head off and I just collect the brain. You could see those sections more distinctly when you do that. But
Okay.
these were in preservatives for a long time, and then they were like in water, washed up and stuff. So it's kind of harder to see. But when you have the blood vessels, like the blood vessels kind of like separate the different section and the
Okay.
brain. Yeah.
Okay. All right. I just knew I wasn't the only one asking that question. I was like, they all look the same color. Thank you.
I
I'm gonna go back
know.
out.
It's fine. Thank you for your question. It was the last thing that I was gonna show is basically the brain slices. Ooh, kind of can't see the brain slices that we use once we stain our gene. So once we have these brain slices, we put it in different chemicals and different fluorescents to target different genes, and then we look at these under the microscope. So that was what I was going to mention. So the cortex would be this area that we're looking at. Because remember, there's a lot of cells, so we don't want to focus in like other cells. We want to make sure that we deleted dopamine in those specific cells that are shown to modulate the type of stress that we internalize. They're located in the midbrain and the prefrontal cortex, which is the cortex is like right here. So that's the last thing I wanted to show. Didn't want to overwhelm you guys. But overall, that's it. I will be taking any questions now.
So we will take our last couple of questions, you guys. I know there was one in the chat that said, your advice for aspiring neuroscientists. What is your advice for aspiring neuroscientists?
It's to keep an open mind, because you can do a lot in neuroscience. I do more of genetic work, but you can do a lot of behavior work. You can do, I don't know if you guys remember what I pointed out, olfactory work, ocular work, looking at the eyes, different like disorders that from people who are blind, and so different stuff like that. So even if you are an aspiring and not don't know what to do, but you are interested in neuroscience, keep an open mind. Be interested in behavior. Be interested in all those different points until you find what you're really, really passionate about. That's very important.
I think that's a really great point. And, also, can you just mention, are there any resources that you know of that kids can get involved in with right now? Like, of course, your company that you've started, I'm sure is a great resource for them. Can you speak more about that and share any others?
Yes, I did not know this when I was in high school, but sometimes you kind of have to be proactive and Google. Look into summer research internships in biology and biomedical and any type of magnet programs. If you qualify for, do them, even if it's studying a lot. Didn't know. I would never know if I was interested in science if I didn't really study it and found that I was interested in like the core classes. Um, so a lot of Googling because you want to not only just study, but have hands-on research in the lab. The sooner you get that over, like started, the better. So resources in terms of internships, there are high school ways to, I've mentored a high school students in my lab in the past. Um, I don't understand and know how he did that, but he did mention that that was an option in high school that he could do a related internship. If you don't find the opportunity, I would advise that you talk to a graduate student such as myself. And you can find my blog is I can't always send that to you. My name is Nella Delva. I'm on Facebook. I'm STEMazing um, and Instagram, and I do have a TikTok. So figure trying to figure that out. Uh, so yes, pretty much Google, be proactive and finding different minority opportunities because they are there. I just didn't know about them. I found out about them when I went to college and I took a full advantage of them then. But it's not too early to learn about research in the lab while in high school or middle school, whichever, because I've mentored students who were in high school before.
All right. I think I can't see if there are any other questions, Tequila, if you see any.
There's one more question. Okay, so what are other behavioral or health changes? I'm sorry, let me try this again. What other behavioral or health changes have been found after deleting the dopamine cells?
That's such a great question. Because we tested for other behaviors as well. For those of you who don't know, I always kind of like use this analogy. If you have a pet, if when your dog is sad now, when your cat is not feeling you. So that's the same way that well, not really the same, but that's the same concept when we test our mice for behavior, because obviously we can't, ask them, how are you doing today? They wouldn't be able to help us with anything. They don't speak our language. But we kind of put them through different like paths. We did a memory test. Basically, if they remember that they've already played with this toy. So the more time they spend with the toy, the more exploration time means that they think it's a new toy. The less exploration time means that they're tired of it. They've been playing too much with it. So that's how we test their memory. They remember that it's a new toy. They don't remember if it's a new toy. We also did anxiety test. The way we do that, and there was no changes, by the way, in memory with the deletion of dopamine in those cells. We also tested anxiety, and the way we test anxiety behavior is that we know that, rodents don't like to be in the light. They like to hide in the corner. So we have this apparatus where there's a section of, corner, and there's a section of like, more open area. So the more time they spend in the open area means that like the less anxiety that they have. That means they're fine. But if they just spend a lot of time in the corner, it means that they're very anxious. They don't even want to try to discover what's going on out there. So there was no changes in anxiety at all. So the only thing that was affected in these mice was how resilient they were in depressive-like behavior. Basically, I just want to put that out there to test how depressed that they are, because they're all mice. We kind of throw them in a bucket of water and see if they're going to swim. So mice that are depressed, um, mice that are
They
depressed, they're
drown?
just, yeah, they sink in the bottom and they don't even want to try to swim. They don't
What?
care to swim at all. So, but a lot of them that had the deletion of that dopamine D1 receptor, they were like, I'm not about to die over here. I'm gonna swim until I can't swim no more. They were like over here trying to make it to the corner, trying to, find a way to escape. So that's how we know, okay, like they're fine. So normally their normal behavior as a rodent is trying to find escape routes. So that's how we kind of establish, okay, so if they don't even try, that means they're just tired of life. They just want to die. But if they do try,
Wow.
it's fine. But yes, overall, there was no other changes in any other behavior that we could find from what we've tested. And there was no difference between male and female as well. So both male and female acted the same.
Wow. So, okay. I just didn't imagine that mice would give up the will to live and just die. Yeah. So, our this has been so interesting. I'm sure we can stay all night because we have a million questions for you, but we won't. So I'm going to do a few housekeeping before I let you say final words. So in the chat, Carlin has put in the survey. So please, whether you watch it live or on our YouTube channel, please make time to complete the survey. Um, Nella mentioned TikTok, so I'm going to put a plug for our TikTok STEM. So if you want to share videos on social media and communicate your science, I'm sure Nella could give great examples on her TikTok channel. We're going to look forward to put it in the chat, Nella.
Okay.
Um,
Yeah, it's
your
amazing.
TikTok, it's
Hmm.
amazing.
Yeah,
And
STEM
so
underscore azing.
just if you follow us on social media, just be sure to look for our upcoming events. So we are going to let you have the last closing words, Nella, with what are your words of wisdom, advice to give to teens about science in general and neuroscience in particular.
Um, so I think my advice is science is hard. I think a lot of the times we associate something that's hard with like you not being able to do it. And then I found out that I did like science, but it didn't mean that science wasn't hard for me. It was hard. It was a hard road to study science, to hard night. But it's just the overall goal that kept me going. I was like, oh, can you imagine like if I were to find the cure for a disorder, be a part of a team that came up with, an effective therapeutic treatment. So that really motivated and kind of fueled my pursuit of this career, really. But it didn't mean that it wasn't hard. Some days I wake up and I just didn't want to have to deal with all the stuff that I have to deal with. So my overall last word would be, even if something is hard, keep going. If you have a goal, if a goal that fuels you and fuels what you want to do, continue with the path. The path is not meant to be easy. Studying is not meant to be easy, but if you're really passionate about the goal of what you can accomplish after you get this degree, then it'll keep you going. And then you'll find a passion with like all the good data that you get, pretty much. That's it.
Sorry. Those were great words. I'm going to let Carlin close us out to keep going, right? Because life can be hard and we're taught to quit, right? So don't be like the mice. Keep going.
Yes, yes. Keep going. And even if you think that you have an issue, we could be depressed, get yourself checked out. As I mentioned, it's not just environmental factors. Sometimes we inherit those genes from, you know, what they call generational curses nowadays, but we inherit a lot from our grandmothers or great great grandmothers, and that dictates how we react to our environment a lot more than we think. And that's important to think about because if we feel like we have a problem, seek help. There's nothing wrong with seeing that something is not normal in our behavior. It is a genetic thing. And there's treatments for that. Get yourself checked out. I mean, with studying depression, there's several times I'm like, okay, I think I'm definitely depressed. I, but I did get myself checked out. I'm fine, but with like reading so much about it and so much it affects the gene and how we respond and how it affects behavior, it just really made me think about other minorities out there that really don't think about it and don't know about that. They just think that, this is the person's character. It could be trauma that they kind of inherit and then change their genome overall. And then their son and their daughters will have the same issue that they have. So get yourself checked out, get yourself treated, and maybe the next generation will be better at dealing with stress overall.
All right. So I really enjoyed tonight's presentation. Nella, you knocked it out of the park with helping us start our new program year and kick us off for the month of August. Like I said, I want to thank everyone for tuning in. Thank you also for those who will rewatch this presentation. It will be uploaded and posted on our YouTube channel. We received our second highest number of registrants for this cafe. So definitely we want to see you at our other upcoming programs. And if you got a lot of value and a lot of entertainment and fun out of this, then you will at our other upcoming programs as well. So our theme for the month is neuroscience and looking at the brain. So our next Teen Science Cafe, which will be in 2 weeks, will also be about the brain, but it will be about memory and the impact of injuries on the brain, concussions. We will also have this month our STEM in the City, the ecology of cultivating food in urban areas. And so that's going to be a really good workshop because I don't know how many of you already have gardens, but if you don't, then this is the workshop that you want to be at to learn more about, producing vegetables and fruits and plants in urban areas. So I have dropped the link in the chat to where you can register for these upcoming events from us. Also, as a final note, we did launch our newest program, TikTok STEM, last week. And I love that Nella mentioned that she has a TikTok account. Definitely follow her. We follow us on TikTok. And I will also drop the link to our launch video so that if you are a teen who loves STEM, who likes to be creative on social media, you can check it out and participate. So I think that's all that I have, Tequila, as far as announcements to close this out.
Okay. Well, thank you for coming, everyone, and good night. See you at our next event, and be safe. Wear your mask, wash your hands, and social distance. Bye.